Hi, my name is Samuel Thompson, and I'm a 2022 Arnold O. Beckman Postdoctoral Fellow in the Chemical Sciences. To celebrate the end of my fellowship, I'm going to tell you about proteins. Proteins are amazing biomolecular machines that biology has used to change the world around us. And I'm going to tell you how we're using proteins to change our world and the future. Let's think about the word protein. I bet you thought of something like this, right? The way most people interact with protein is through nutrition. You're probably also thinking about something that's just sitting there, either on a store shelf or on your plate. When a biochemist or a biophysicist thinks of protein, we're thinking a billion times smaller, nanometer scale. We see molecules in motion. We see machines carrying cargo or chemically changing other molecules. We see the essential machinery of life in proteins, all the tiny parts that keep the cells in your body functioning. That all happens through proteins. And we use them in our food to turn milk into cheese. We use them in laundry detergent to get stains out. And now we even use them as medicines to target everything from migraines to cancer. But there's one common factor in all these applications. The proteins are being used in water. That's fine for many things. There's certainly a lot of water in our world, but there's a lot of parts of our world where there is no water, or there's places where water can't go. So I want you to imagine what would happen if proteins worked in places other than in water. Take a moment. If we understand the biophysical principles to design proteins specifically for unusual environments without water, first, we could expect to have more stable, long-lived industrial catalysts. For example, if we could convert biodiesel from the 3 billion gallons of waste cooking oil produced in US restaurants alone, we could offset about 7% of US diesel consumption. Second, we could develop biotechnology that interfaces with natural hydrophobic environments, such as cytosolic lipid droplets, to develop novel therapeutics for viral infection, obesity, and cancer proliferation. Third, we could use self-assembling proteins to build self-healing membranes for long-lived lithium-ion batteries, or to build nanoscaffolds for 3D semiconductor chips. And we can even ask planetary questions to imagine what would life be like and what would biomolecules be like in radically different environments, such as the methane lakes on Titan. So how do we get proteins that do all of these amazing things? When you pour a glass of water, all the molecules stay together. For water, the dominant force is polar interactions. Positively and negatively charged atoms share bonds with a single hydrogen, so we call these hydrogen bonds. For oil, the dominant force is nonpolar interactions. The cloud of electrons around two molecules overlap and cause them to stick together a little bit, and we call these van der Waals interactions. But we also know that oil and water don't mix. Polar and nonpolar molecules don't make strong interactions with each other, so they tend to associate with themselves and form their own preferred interactions, hydrogen bonds and van der Waals interactions. This causes them to separate. So now imagine what happens if a molecule is part oil-like and part water-like. To think about this, I want you to imagine a long molecule, like a chain or a string of Christmas lights. And on that chain, there are oil-like parts and there are water-like parts. If we put that chain into water, the molecule will start to collapse and fold up like origami. And in its most compact and folded state, it will have formed a structure of folded elements connected by loops. And the center, or the core of that structure, will be this little drop of oil that's hidden from water, and the outside will be polar and form favorable interactions with the water, including hydrogen bonds. So now, what if you want to make a protein that leaves water to work in industrial chemical environments? We think you can do this by switching the architecture of proteins to put polar hydrogen bond interactions in the core and putting the non-polar van der Waals interactions on the surface. First, we design a protein with a computer using engineering modeling software. Then, we encode the design sequence into DNA so we can make it. And we make that protein in a test tube reaction that has both water and oil. And we wait to see where does the protein end up. Does it end up in the water or does it end up in the oil? This ability has been transformative for our experiments, but we didn't stop there because we think big. And so in order to think big, we thought really, really small. What we've done is we've created what's called a microfluidic droplet. That's a droplet that is generated by these small devices that flow liquids through them to make complex geometries out of different liquids. And what we get out of that is we get basically a test tube that is a tiny, tiny fraction in volume of a normal test tube. And we can run reactions in those little test tubes. The other thing that's really amazing about these is that we can use commercial machinery to analyze these at rates of millions per hour. So now we have a platform that allows us to create huge amounts of information and run millions of experiments at once using very, very little material. It's an incredibly powerful tool that we think is going to reshape our ability to discover proteins that do the weird and unusual things that we want them to do. That is the legacy of Arnold O. Beckman, creating tools that allow researchers to pursue exciting and transformative science. The Arnold and Mabel Beckman Foundation enables young researchers like myself to establish our careers by building the tools that allow other researchers to tackle the most challenging problems in science. Together, we are creating new avenues for progress. I'd like to thank the Arnold and Mabel Beckman Foundation for their visionary support of this project. We would not have been able to get through the hardest problems here if it hadn't been for their continual support and their generosity. They've really made everything possible. So thank you for continuing the mission of the Arnold and Mabel Beckman Foundation, and thank you for your time.